Good morning, biology. Today we're going to go through the cell cycle. This really is the story of how you went from one cell big to many cells big. So if you remember back to the beginning of this unit, that was kind of the overall theme of this unit, is that you started as a single-celled organism. You yourself did, right? When sperm cell met egg cell, that very first cell that you were, when you were just one cell big microscopic organism, you've been copying the DNA inside that first original cell, and we've talked about how DNA replication works, and then moving copies of that DNA to opposite sides of that first original cell, and then you just split that cell in two, and now you were 2 cells big. And then you repeat that process and you became 4 cells big, and you repeat that process and you became 8 cells big, etc., etc., etc. And you went through all these phases of development where you just added more and more and more cells. And really what you are now is just 100 trillion cells all stuck together. That's how our story goes, from one cell big to many cells big. The steps of going from 1 cell to 2 cells, that's the story we're going to tell today. So today, learning target D2, we're going through the cell cycle and the steps of the cell cycle. The cell cycle is, not only does it tell you how you grew up, how you went from 1 cell big to many cells big, but also if you damage yourself, like let's say you, get a paper cut or something, how you're going to fill in that paper cut is by growing new cells. So your body still is making new cells all the time. Every time you move or do anything, cells are falling off of you. Cells wear out and get destroyed, and then your body has to get rid of old dead parts of cells and then regrow new cells. So you're constantly doing this all the time. It's just that now, when you were little and you were growing, you were adding cells. You were doing the cell cycle faster than cells were wearing out. But now you're doing it at about the same rate. So as cells wear out, you replace them. So your cells, the important take-home point is that your cells are still going through the cell cycle, and they'll never stop your whole life. Understanding how things go wrong in the cell cycle, and we talked about this a little bit when we hit on DNA replication, but understanding things that go wrong with the cell cycle will make you understand how cancer works. And it's kind of important to understand how cancer works because the leading cause of death in normal times when there's not a pandemic, the leading cause of death globally is heart disease and cardiovascular disease. So your blood tubes clog up and give you either a heart attack or a stroke. That's what's going to kill 70% of people on the earth. But then the next leading cause of death is cancer. Around 20% of people are going to die of some form of cancer. So really common, and odds are pretty good that you yourself or people you know have had experience with cancer. It's that common of a disorder. So understanding the cell cycle is the key to understanding how cancer actually works. Because really what cancer is loss of control of the cell cycle. It's your own cells. A cancer tumor is your own cells reproducing out of control. So this is a pretty important topic to understand today. I guarantee you that if you haven't had personal experience with cancer yet, you will at some point in your life. Either you or a loved one is going to battle cancer, and that's kind of spooky. You should at least understand how it works. Cells have a life cycle that contains 5 stages. So we're going to go through those 5 stages right now. That's the goal of today, is that you understand all 5 stages of the cell cycle. Here we go. Step 1 is going to be a phase called G1. Which I like to think that G1 stands for growth 1, because that's the main thing that happens in the first step of the cell cycle, is that your cells just grow. So a young cell is going to grow by adding parts and absorbing organic molecules from its surrounding to build more parts. So your cells are surrounded by blood, okay? So like cells in your body right now have a blood supply coming to them. Your blood gets loaded up with all the stuff your cells need to grow and repair. So every time you eat a meal, all of those molecules from that meal, all the fats and carbohydrates and proteins and nucleic acids that came in from the meals you eat, that all goes diffusing into your blood. And from your blood, all those organic molecules go diffusing into all of your cells. And your cells are going to use those organic molecules to add parts to themselves. So your cells are getting bigger and they grow. We talked about surface area to volume ratio. There's a limit to how big cells want to get. When their surface area to volume ratio gets too low, the cell's going to need to divide to keep diffusion going fast enough. So that's kind of the whole idea for what triggers us to go through the steps of the cell cycle, is that cells have a limit to how big they can get. New building blocks for building your cells are going to come from the molecules in the food you eat. You literally are made out of what you eat, because we've talked about matter doesn't just come from nowhere. Like when you grow or get bigger, there's no magic. Like that matter had to come from somewhere, and that matter is coming from the food you eat. All food you ever eat came from another living thing, and all living things are made out of proteins, fats, carbohydrates, and nucleic acids. So that's where you get the building blocks to build you, is from other living organisms that you eat. Okay, there you go. Just to refresh your memory on what cells are built out of. They're built out of carbohydrates, proteins, nucleic acids, and lipids. Those are the 4 types of molecules that living things are built out of. So when you're in the G1 phase of the cell cycle, and this little diagram over here is going to be showing us the steps of the cell cycle. When we're in this first part of the cell cycle, like let's say I'm a brand new cell, like if I was that first cell that happened when sperm cell met egg cell, if I was a brand new cell, I'd add parts to myself through this G1 phase by just absorbing these molecules from the food I eat and growing and adding parts to myself. Okay, that doesn't seem too complicated. G1's pretty simple. It's just a growth phase. The G actually stands for gap, for reasons we'll get into later, but I always like to think of it as standing for growth, because the G1 phase is the growth phase where cells add parts to themselves. Second step is going to be the S phase. All right. I wish they would have called it the R phase, because what we're doing is DNA replication, and replication starts with an R. But another word for replication is synthesis. Synthesis means making stuff. What we're making during this S phase is a copy of our DNA. This is where we do the whole DNA replication trick that we covered yesterday. So this is where, in this phase, this is where we're going to split our DNA, unzip it like a zipper, and then line up new DNA nucleotides on both sides of our old DNA strand, making 2 strands of DNA. So at this midlife point in a cell's life, it's already copied its DNA. That's what happens during the S phase. And the cell continues to grow and add parts to itself and do its daily cell job. So if it's a liver cell, it's doing liver jobs, or if it's an eyeball cell, it's doing eyeball jobs, but it's also copying all of its DNA. Remember that DNA is found in little packages, or I shouldn't say it's a package. It's not like the chromosomes contain DNA. Chromosomes are made of DNA, right? So the chromosomes go from looking like a stick to looking like an X, because really what's going on when we talk about chromosomes, I'm going to use that word a lot, so it's pretty important we understand what chromosome means. If you think of your DNA as sitting around inside a nucleus, your DNA is actually sitting around in 46 little packages that look like this. And if I unwound this package, it would look like a noodle of DNA like that. But that noodle of DNA is all packed up into kind of this worm-looking structure. This is just wadded up DNA. We would call this a chromosome, or sometimes you see this called a chromatid, T-I-D. Really, how I like to refer to them as stick-shaped chromosome. This is a chromosome when the cell is in the G1 phase. So if I open up the nucleus of the cell here, what would come spilling out at me is 46 sticks of DNA that look like this. And if I unravel this, it would look like that twisted ladder shape. Okay? After S phase, I have 2 copies. So I have 2 sticks that look like this. But really, what DNA does, for weird reasons that I don't really want to get into right now, your 2 sticks of DNA stay stuck together in the middle. So you end up with a structure that looks more like, I meant to erase that. There, erase that. You end up with 2 sticks that stay stuck together in the middle, and you end up with something that looks more like this. Ta-da! This X-shaped structure, that's actually 2 identical chromosomes, 2 replicated pieces of DNA stuck together in the middle. So anytime you see a structure that looks like this, that's really 2 copies of the same DNA. So if I drew like a dotted line down the middle, like this side is 1 copy, this side is 1 copy, and they're identical to each other. The 2 sides of this chromosome are identical to each other. The chromosomes would be X-shaped like this at the end. In other words, they have 2 copies stuck together in the middle by the end of S phase. At the beginning of S phase, like right when we come out of G1, we would just have the 1 stick of DNA. So looking more like, right? Ta-da, 1 copy of DNA. coming out of G1. By the end of S, we've got X-shaped chromosomes. So don't get confused by that. When I start using that terminology, stick-shaped chromosomes, X-shaped chromosomes, I'm talking about, is the DNA copied or not copied? If it's not copied, it's stick-shaped. If it is copied, it's X-shaped. At the end of the S phase, 2 complete sets of DNA will be in the nucleus of each cell. So in other words, at the end of S, the cell has twice the DNA that it actually needs. It's preparing to split. Your cells are preparing to split. They're doubling up their instruction book so that they can give their offspring an exact copy of your genetic instruction book, your DNA. That's a prettier picture of what I just tried to draw. So on the left there, we've got an unduplicated chromosome. And on the right, we have a duplicated chromosome. Some textbooks call an unduplicated chromosome a chromatid. And then when it's duplicated, they call that a chromosome. A chromosome is just made of 2 identical sister chromatids. If you're not good with that, you can email me questions and stuff. I'll be happy to explain that further, but that's what's going on with duplicated versus unduplicated chromosomes. At the beginning of S phase, it would look like the picture on the left there, and at the end of S phase, it would look like the picture on the right. Ta-da. Okay, step 3. The G2 phase. Well, in G1, cells just grew, right? They just added parts to themselves. And then in S phase, we copied the DNA. In G2, it's another just kind of hang out, do your daily cell job, and grow some more. So in this step, the cell grows and develops some more. It's also going to add organelles. So remember, it's not just the DNA we need to worry about making twice as much of. We got to worry about making twice as many mitochondria, twice as many ribosomes, twice as many endoplasmic reticulum, twice as many Golgi apparatus, all the parts that we learned about that are inside cells. We got to double all of that stuff if we're going to divide the cell, because each offspring cell is going to need all the organelles it needs to survive. So it doubles up on everything. By the end of G2, this cell is getting really uncomfortable because it's getting so large because it has to make double of everything. And remember, cells don't like being large. It makes their surface area to volume ratio small. And so by the end of G2, this cell is very uncomfortable. Diffusion is slowing down. It can't keep itself going like this. It's going to need to divide soon. So there you go. By the end of G2, the surface area to volume ratio of the cell has gotten too small because now our cell is way too big. It just can't keep operating like this. But it's doubled everything. This is like a cell that's twice as big as it needs to be by the end of G2. All right, so that means those last 2 arrows there, the last 2 arrows that we haven't filled in yet on the cell cycle, those are going to be the dividing stages. Those are going to be the stages where the cell starts doing the processes that it needs to do to split in 2 and become 2 offspring cells, or what in biology, the default gender they use to describe things is female. So they call them daughter cells. The one parent cell is going to split and become 2 daughter cells. That's what's going to happen during those last 2 phases. So let's look at those last 2 phases. Step 4 is a process called mitosis, which we're going to zoom in on tomorrow and start looking at it in greater detail. This is where we're going to take our X-shaped chromosomes, our 2 copied sets of DNA, and we're going to move them to opposite sides of the cell, getting ready to split the cell down the middle. So this whole process of mitosis is complicated, and like I said, we're going to go through the steps of how we separate one of those X-shaped chromosomes into its sister chromatid components into 2 stick-shaped chromosomes, and then we're going to move those 2 sticks of DNA to opposite sides of the cell so that each new baby cell gets identical copies of DNA. So by the end of this step, by the end of mitosis, all the DNA for one complete copy of DNA has been moved to the left side of the cell, and 1 complete copy of DNA has been moved to the right side of the cell. And now our cell is ready to divide down the middle, and each daughter cell is going to receive an identical copy of that original set of DNA. The final step, that little C step there, is called cytokinesis. This is like cellular karate chop. You're going to cut the cell in half. So during this step, the cell membrane pinches together in the middle. The cell separates into 2 identical cells, and each has an identical copy of DNA. And each has copies of all the organelles that we copied during G2. So each offspring cell now is just like the original parent cell. So there's a little diagram of kind of how the overall process would work in still shots. We'd start as one cell big, and it would divide. All the identical copies of the DNA and the nuclei would go to opposite sides of the cell, and then we split the cell in two. Boom, we've got two cells where we used to have one. And notice the two cells on the right side of that little diagram at the bottom look identical to the one cell at the left side. Or here's a little video of cells doing the whole cell process. Here's cells dividing. That was one cell becoming two cells. Oh, there's one cell becoming two cells. There's one cell becoming two cells. I really like that one. One cell, two cells. And you've just been doing this over and over and over again since you were conceived, since sperm cell met egg cell. This is how you went from one cell big to many cells big. Now those two cells, every time one of these gets done dividing, those two cells are going to go through the stages of the cell cycle again. So those two offspring daughter cells are going to grow and develop during G1. They'll copy their DNA during S. They'll grow some more during G2. They'll move their copied DNA that they did in the middle of their life. They'll move the copied DNA to opposite sides of the cell during mitosis, and then finally the cell divides during cell division. One thing that people get wrong often on the tests and stuff over this is they'll tell me that mitosis is where we copied DNA, and that's just plain not true. Mitosis is where we moved the copies of DNA to opposite sides of a cell. Where we copied the DNA was during S phase. So I always think of it as like, when humans have a midlife crisis, they buy a stupid sports car, says the guy who drives a stupid sports car. But when cells have a midlife crisis, they copy their DNA. So that's stupid. But that will help you remember when do we copy the DNA in cells? Well, they copy it in S phase, which is like right in the middle of their life cycle. They move those copies to opposite sides of the cell during mitosis. Okay. Tomorrow, we're going to start zooming in on mitosis. Interphase. I forgot to mention this. This is important. These phases right here, I will highlight them in a color. The ones where the cells are just hanging out doing their normal everyday job. So like if I'm a heart cell, I'm doing just heart things. I'm contracting over and over again to make the heart beat, okay? So if I'm a heart cell, I spend most of my time just doing my heart cell job. Like, don't forget, all these cells have jobs to do to make your body work. This whole section right here, the section included in not the dividing parts, like mitosis and cytokinesis are the parts where cells divide. All the rest of the cell's time is spent doing its normal everyday job, as well as doing the growth and development in G1 and copying the DNA in S, and then doing more growth and development in G2. Yeah, they have to do all that stuff that's involved in the cell growing and getting bigger, but they also have to do their normal everyday cell job. So if I'm an eyeball cell, I got to do eyeball things, or if I'm a heart cell, I got to do heart things, or if I'm a liver cell, I got to do liver things. So cells doing their everyday job, we call that a cell that would be in a stage collectively called interphase. So I want you to know that word too. If I were to ask you on a test or quiz or something like what's included in interphase, you would tell me all the steps except for the dividing steps. G1, S, and G2. That's all interphase. Okay. Make sure you know what interphase means. If I randomly plucked a cell out of anywhere in your body, chances are that cell would be in interphase. It'd be very rare to catch a cell that was in the middle of the dividing steps in mitosis or cell division. If we had time, and if this was a normal year, we'd take your cells out and look at them under a microscope. We scrape them out of your cheek and then look at them under a microscope. Most of your cells would be in interphase. Okay, tomorrow we're going to zoom in on mitosis and cytokinesis, and I'll talk about how cells actually do the dividing steps because that gets a little bit more complicated. So mitosis and cytokinesis is the theme for later on this week. I don't know if we'll get to that tomorrow. I think I'm going to have you work on the reading guide for this unit starting tomorrow, or maybe even starting today. I'll post it up today if you want to get an early start on it. I'm having you do the reading guide, getting ready for your unit test, which is coming up on Friday of this week. So all Learning Target D stuff, I could test you over on Friday of this week. So please make sure that you're up to date. If you missed a video or you missed like yesterday's lecture or anything, please make sure you go back and take notes on that. Just a reminder, you should be taking notes and keeping up with class every day. If you're falling behind or you're struggling with something or you're not understanding something, please email. I'm available via email anytime. That's all for today. Email if you got any questions. Talk to you later.